Right. Along with neuroanatomy, we will try to discuss about embryology also side by side. When you are studying about embryology, we should know some basic concepts regarding what is growth, what is differentiation, what is cell division. In that there will be two types, mitosis and meiosis. From there we will go for directly embryology. First I will give these basics. Today, what is growth, what is differentiation and what is cell cycle and mitosis. Those things we will discuss now. First, if you take growth, what is growth? Increasing the size of the organ and organism. Is it or not? Increasing the size, what we call growth. This growth will occur under three processes. What are those? First one is multiplicative growth. Multiplicative growth. Multiplicative growth. Second one is oxitec growth. Oxitec growth. And third one is accretionary growth. Accretionary growth. Right? Accretionary growth. First, if we take multiplicative growth, in word itself meaning is there, multiplication. One cell undergoes multiplication or cell division, produces two cells. Then these two cells undergoes mitotic division and produces four cells. These four cells again undergoes cell division and produces 8 cells. So what is happening here? Increasing the number of cells so that growth will happen. That type of growth what we call multiplicative growth, and this type of growth is most common. Clear? Then oxitec growth. Oxitec growth means what? Instead of increasing the number of cells, here cell size will increase. Size of the cell will be increasing. So that type of growth what we call oxitec growth. Where can you find? We can found in the larger neurons and conversion of oogonia into oocyte, primary oocyte. At that places, size of the cell will be increased. Right? So just imagine this is the cell. When it undergoes oxitec growth, this cell increases its size. Right? But here we have to discuss one thing. If smaller cell, it will have sufficient nutrition and it can produce by its own. But if it is larger cell, it cannot produce sufficient nutrition by its own. Right? For that, it needs some external nutrition from external source. So those type of cells are surrounded by some supporting cells. Those supporting cells, in case of oocyte, follicular cells. You must be aware of this word, follicular cells. Right? So these are follicular cells. These follicular cells gives nutrition to this oocyte. Clear? In case of larger neurons, like neurons which are present in the dorsal root ganglion, those neurons also larger cells. Those larger cells are surrounded by satellite cells and fibroblast cells and forms capsule around the neurons. is it? So this type of growth, that means if the cell size increases, that type of growth what we call oxitic growth. Then accretionary growth. Accretionary growth means instead of cells, interstitial substance will increase, increasing the interstitial substance, increasing the amount of interstitial substance, right? Examples, cartilages and bones. There interstitial substance will be more and those structures will undergo growth. That type of growth what we call accretionary growth. Clear? So these are the different types of growths. Then what is differentiation? Differentiation is nothing but, I am removing this. Differentiation is nothing but it is a process in which cell assumes specific characteristic features and assigned for special functions. Very simple. This is a process. In this process, cell assumes some special characteristic features. Because of that special characteristic features, that cell can do some special functions. Imagine, first stem cells will be there. These stem cells undergoes differentiation and produce different types of cells like neurons, that one type of cell, liver cells, cells which are present in the skin and cells which are present in the blood. They will have different functions. Neuron functions are different, conducting the impulses. Liver cell function is different, production of bile, right? And pancreatic cell function is different. Of course, there will be two types will be there. Two types of cells will be there: exocrine cells and endocrine cells. Exocrine cells will produce pancreatic juice and endocrine cells will produce insulin and glucagon. So that process what we call differentiation. Let me tell you once again. Just imagine this is stem cell. This stem cell undergoes differentiation and it produces different types of cells. Those different types of cells assigned for specific function, right? So that is differentiation. And what are the different types of stem cells? Just see here. This is first type of cell is totipotent cells. Totipotent cells, right? These totipotent cells can produce any type of cells, either extra embryonic or intraembryonic. Example is embryoblast. These cells, they can produce any type of cells, right? Like they can give contribution for the placenta, they can give contribution for the fetal membranes, and they can give contribution for the fetus also, right? So those type of cells what we call totipotent cells. Example for these totipotent cells are embryoblasts, right? That means initial cells of embryo, right? Then pluripotent cells. Pluripotent cells. Pluripotent cells. Pluripotent cells means what? They will be derived from the totipotent cells, but they cannot produce all type of cells, but they can produce most of the cells. Example for this is inner cell mass. Inner cell mass, right? So inner cell mass can produce fetus, right? But it cannot produce placenta and fetal membranes, is it? But totipotent cells they can produce. Clear? So here these totipotent cells undergoes differentiation and they can produce pluripotent cells. Why I am drawing like this? They can divide and they can produce pluripotent cells and they can also produce totipotent cells. Clear? So that is about totipotent cells and pluripotent cells. Right? Then next, multipotent cells. Multipotent cells, of course they will be derived from the pluripotent cells. Multipotent cells. These multipotent cells derived from the pluripotent cells and they will undergo differentiation and they can produce different types of cells. That means multiple type of cells. Multiple type of cells means what? If you observe blood stem cell, it can produce RBC, it can produce WBC, it can produce platelets. Right? From the same stem cell. What is that stem cell? Blood stem cell. Right? So next cell is multipotent cells. Multipotent cells. Right? So these multipotent cells can produce different types of cells. Just imagine this is blood stem cell. Blood stem cell. They can produce RBCs, WBCs and platelets. And of course they can produce stem cells also. Right? Now from this cell there will be another type of cell. That type of cell is what we call unipotent cells. Unipotent cells means they committed to produce only one type of cells. Not like this. This can produce RBCs, this can produce WBCs, they can produce platelets. But unipotent cells committed for single type of cells. Just take an example, neuroepithelial cells. That means neuroblasts. These neuroblasts, that means nerve stem cell. If you take this is nerve stem cell. Nerve stem cell. Nerve stem cell. These nerve stem cells can produce only neurons. They cannot produce any other type of cells. Right? So this type of cells what we call unipotent cells. Unipotent cells. Unipotent cells. Clear? So that is about differentiation. Clear? I hope you understood what is growth, what is differentiation. Now we will go for our topic proper, cell cycle. Right? Right. Now we will discuss about cell cycle. Cell cycle consists of two phases. I hope you must be aware of it. Interphase and M phase. So this is interphase. This is interphase. Right? Then next phase is M phase. This is M phase. So right? So this is interphase and this is M phase. Interphase further divides into G1 phase, S phase and G2 phase. So here I am drawing like this, so that this is M phase, this is G1 phase and this is S phase and this is G2 phase. Clear? Now if you take one cell, it has to pass through all these stages, then only cell divides into two cells. Clear? Now if you take one cell which contains cell membrane, within the cell membrane we can find protoplasm, right? What is protoplasm? What is cytoplasm? Protoplasm is nothing but total content which is present inside the plasma membrane or cell membrane. That material what we call protoplasm. Then what is cytoplasm? Very simple. Protoplasm minus nucleus. If you remove the nucleus from the protoplasm, that total content what we call cytoplasm. Cytosol means the material which is present in between the organelles, right? That means what? If you remove the all organelles along with nucleus from the protoplasm, the remaining material is cytosol. Clear? Right. Now this cell is entering into cell cycle. Once this cell enters into cell cycle, first phase is G1 phase. Here G stands for gap, but here growth will occur. What is that growth? First, increases the size of the cell. Size of the cell will increases, right? So here size of the cell increased. Clear? Then there will be accumulation of materials which are necessary for the synthesis of DNA and synthesis of organelles, which will happen in the G2 phase, and division of cell. For all those processes some materials are needed. Is it or not? Right? Some enzymes, proteins, all those things will be needed. So those materials will be accumulated within the cell in G1 phase. Clear? So that is about G1 phase. Then S phase. Once G1 phase completes, it enters into S phase. Clear? So what will happen in the S phase? S stands for synthesis. What is going to synthesize here? DNA. Synthesis of DNA happened in the S phase. Why there should be synthesis? See here, this cell is going to become two cells, right? At the time, how many DNAs are present in this cell? Anyone? Okay. How many chromosomes are present in the cell? That you can tell. 46 chromosomes. Is it? Now, if I explain about organization of chromosome, then you will come to know how many DNAs present in the cell. Right? Let me tell you about that. If you take double stranded DNA, this is double stranded DNA. Right? This double stranded DNA winds around histone proteins. Imagine these are histone proteins, right? This DNA winds around histone protein and forms the small portion what we call nucleosome, right? Of course, this is another nucleosome, right? But this total material, this material what we are calling chromatin filament. This is totally what we are calling chromatin filaments. These filaments again coiled and forms chromatin threads. Of course, I didn't draw the histone molecules here, so you can put the histone molecules, right? So these chromatin filaments coiled again and forms chromatin threads. These chromatin threads further coiled and forms structure like this. Now you tell me, how many DNAs are present in the single cell? Very simple, 46 only. Clear? When in resting cell. But if you observe, after synthesis phase, in this cell you can observe double number of DNA. That means 46 plus 46, 92 DNAs will be there. How they will be, I will tell you. Clear? Now this cell contains 46 chromosomes, but this cell undergoing cell division. When it undergoes cell division, daughter cells also should have 46 chromosomes. Is it? Actually, daughter cells are identical to mother cell during mitosis. Is it? If it is identical, how many chromosomes which are present in the mother, same number of chromosomes should be present in the daughter cells also, right? So that's what here DNA will be replicated, so that one DNA becomes 2 DNA. At that time when cell undergoes cell division, these chromosomes will be equally distributed. Clear? Right. Now if you take single chromosome, one chromosome before synthesis phase, you can observe like this, right? When it condensed, when the chromosome condensed before synthesis phase, chromosome appears like this. But if you observe chromosome after S phase, when it condensed, why I am stressing this word condensed? If there is no condensation of these chromatin threads, you cannot observe chromosome like this. Just you can observe like chromatin threads, right? When it condensed, then only you can observe like this. Clear? When in resting condition, right? But when it completes its synthesis phase, at the time chromosome appears like this. So what does it mean? This is single chromatid. This chromatid contains one DNA. Here this is single chromosome, but two chromatids are there. This is one chromatid and this is another chromatid. This chromatid contains one DNA and this chromatid contains another DNA. So these chromatids what we call sister chromatids. This is one sister chromatid and this is another sister chromatids, right? Understood? So before synthesis phase, chromosomes will be like this. After synthesis phase, chromosomes becomes like this, right? That too when it condensed. Actually in G2 phase it will not condense, in prophase only it is condensing, right? But for your understanding purpose I am telling like this. Clear? I hope you understood why there should be synthesis. Clear? So here synthesis of DNA. Clear? Once synthesis completes, this cell undergoes G2 phase. Once it enters into G2 phase, there will be multiplication of organelles, cell organelles. You must be aware of it. Cell contains different organelles: endoplasmic reticulum, Golgi complex, ribosomes, lysosomes. All these structures will be there, right? So all those structures here doubled. That means organelles of the cell also become double. Why? This cell is going to divide into two. Organelle should be sufficient for two cells. So here growth will take place. That growth is multiplication of organelles. Like division of family only. When one family is going to divide into two family, at that time they will arrange two sets, everything, right? Then they will divide. Same thing will happen here also. Clear? Then once G2 completed, it will enter into M phase. This phase what we are calling M phase, right? M stands for mitosis in case of mitosis, but this M stands for meiosis in case of meiosis, right? So in meiosis also cell cycle will be there. Clear? So I hope you understood up to G2 phase, right? Now we will go for M phase. In M phase there will be different types of subphases are there, that we will discuss one by one. Can I remove this? Right. You must be aware of different phases of M phase. That means mitotic phase, prophase, metaphase, anaphase, telophase. Right? Prophase, metaphase, anaphase, telophase. Right? Then how will you remember P, M, A, T? Actually we should remember in order. Prime Minister's America Tour. Remember like that. Prime Minister's America Tour. Prophase, metaphase, anaphase, telophase. Clear? Then first we will take prophase. Here for explaining prophase, I am taking this cell after completion of G2 phase. I am taking this cell here, right? But here when I am taking this cell, one more thing you have to remember here. See, different types of cells are there. Squamous cells are there, they are flattened cells. Cuboidal cells are there, they are cubical in shape. Columnar cells are there, they are elongated cells. Polyhedral cells are there, they are in polyhedral in shape. Right? Umbrella shaped cells are there, they are like umbrella shaped. Is it? Those cells, when they are undergoing cell division, just imagine squamous cell is there. If it is flattened cells, during cell division there will be so many processes will take place, like formation of spindle, contraction of spindles, and segregation of chromosomes. Like this, so many processes will take place. For that process, cell needs some space, right? That's what in G2 phase itself there will be one more important process will take place. That process what we call cell rounding. Cell becomes round, right? So that process what we call cell rounding. Clear? So after cell rounding I took the cell, right? Now This cell is in prophase. This cell is in prophase. This is prophase. And what is the main change will occur? Condensation of chromosomes, so that it appears like this. Single chromosome looking like this. So what is this? Pair of chromatids are there. Pair of chromatids. So here prophase, P stands for paired up. P stands for paired up. Clear? Now this is prophase, first step of M phase, and which contains nucleus. This is nucleus, right? Then remaining all other organelles will be there, but I am taking whatever the necessary organelles for our explanation, right? Then here nucleolus is there. Clear? Then chromosomes, but these chromosomes we cannot see like that in this phase. Just it will be like threads. It looks like just threads, right? Now one more thing I have to draw here. Those are centrosomes. So these are centrosomes. Usually cell contains only one centrosome, but in G2 phase this centrosome also multiplied and became 2. Clear? Now we will see one by one, step by step, what are the different changes which occur in the prophase. First, you should strongly remember that it is disorganizing phase. It is disorganizing phase. Clear? So what will happen first? Condensation of chromosomes. Condensation of chromosomes. When these chromosomes condensed, appearing like this. So condensation of chromosomes. Now these chromatin threads condensed and formed like this. Right? I am taking 2 chromosomes here. I never mean that this cell contains only 2 chromosomes. Right? 46 chromosomes will be there, but I cannot draw 46 chromosomes in that small nucleus. Right? That's what I have drawn 2 chromosomes. Clear? Then, and you can ask me one more thing. Why DNA is winding around histone protein, then these filaments, that means these chromatin filaments again winding and forms chromatin threads, and again these are winding and forming the chromosomes. Because what is the size of the nucleus? Just after all around 6 microns. Then what is the length of DNA, single DNA? It is around 1.8 meters. Right? So 1.8 into 46. Just that means this is 1 chromosome length, like that 46 chromosomes will be there. So see how much length of DNA should accommodate within this smaller nucleus. That's what it will be winding, rewinding and super winding. Clear? Right. Now what is the first step in the prophase? Condensation of chromosomes. Clear? So it appears like paired chromatids. Right? So paired chromatids are formed. Then what are these 2? centrosomes. These centrosomes start organizing to produce microtubules. So here production of microtubules, right? So when they produce microtubules, these microtubules are there, no? Microtubules producing from this centrosome and microtubules producing from this centrosome, both will push us against each other and growth. When they are growing, they will be pushing against each other, so that these structures will move towards the poles of the cell. Length of the microtubules increasing and migration of centrosomes. So these centrosomes will be migrated here. So here one centrosome, here another centrosome, right? Clear? Then along with, what are these? Microtubules. Of course, there will be different types of microtubules will be there. Clear? First one, kinetochore microtubules. Kinetochore means what? It is microtubular binding protein molecule present over the centromere. Over the centromere, there will be microtubular binding structure. That microtubular binding structure, what we call kinetochore, right? So some microtubules, they are made for attachment of kinetochore, right? So these type of microtubules, what we call kinetochore microtubules. These type of microtubules, what we call kinetochore microtubules. Those kinetochore microtubules attaches to kinetochore. That's it. Then next microtubules are polar microtubules. Polar microtubules means, just imagine this is one polar microtubule and this is another polar microtubule. This is one polar microtubule and this is another polar microtubule. Imagine this is one polar microtubule and this is another polar microtubule. They will search for each other and they will interact with each other and they push against each other, so that elongation of cell. How they will push against each other? This microtubule also growing, this microtubule also growing. So when they are opposing with each other, size of the cell increases, right? So that elongation of cell elongates, elongation of cell, right? So that is about which microtubule? Yes, polar microtubules. Then next, another type of microtubules. These microtubules, what we call anchoring microtubules. Anchoring microtubules. What does it mean? What is the function of it? Anchoring, supporting. Why this cell needs support here? Here, two microtubules, polar microtubules, pushing against each other, so that cell is elongating. And in the next stage, these kinetochore microtubules attaching to kinetochore and they pull, right? So here some microtubules are pushing, some microtubules are pulling, right? So if there is no any support to this aster, actually this total structure what we are calling aster, if there is no any anchoring or if there is no any support to this, what will happen? It will displace within the cell, right? That's what some of the microtubules attaches to cell membrane. These microtubules what we call anchoring microtubules. Clear? Right. Now, after formation of aster, there will be disappearance of nuclear envelope. Disappearance of nuclear envelope. See here, this nuclear envelope will be disappeared. Disappearance of nuclear envelope. How? By phosphorylation of lamins. By phosphorylation of lamins which are present underneath the nuclear envelope. Those lamins phosphorylated and this nuclear envelope fragmented into membrane vesicles. Fragmented into membrane vesicles and remain within the cell. Why? These vesicles are helpful for the reappearance of nuclear envelope. That means reformation of nuclear envelope. I will tell you that in the last phase, telophase. Clear? So now nuclear envelope disappear. Once the nuclear envelope disappear, these kinetochore microtubules enters into nuclear space. Enters into nuclear space. They enters into nuclear space and attaches to kinetochore right. So they are going here and they are attaching to kinetochore, right. They are entering into nuclear space. Of course, some algae and fungi, this nuclear envelope will not disappear. Just it will be present like that only. These microtubules will pierce the nuclear envelope and attaches to kinetochore of chromosomes. That type of cell division or that type of mitosis, what we call closed mitosis. Closed mitosis. But here nuclear envelope disappearing and microtubules are entering into nuclear space. Right? So this type of mitosis what we call open mitosis. This type of mitosis what we call open mitosis. Clear? Now this is open mitosis. Now aster form, nuclear envelope disappear and disappearance of nucleolus. This nucleolus is there, no? This nucleolus disappeared. Right? Clear? So these are the changes which occur in the prophase. Very simple. For explanation it took too much time. I will tell you very simply. Condensation of chromosomes, formation of aster, disappearance of nuclear envelope, disappearance of nucleolus. That's it in the prophase. Then at the last phase of prophase, these microtubules, what are these microtubules? Kinetochore microtubules. These kinetochore microtubules start contracting. See here, imagine this is one thread. If you pull this thread, what will happen? It will come in the center, right? So here also we are pulling, here also we are pulling. So chromosomes will arrange in the center. That will happen in the metaphase, right? Let me draw next diagram, that is metaphase. Here this is the cell. This cell contains chromosomes in the middle. Why? Because of contraction of these kinetochore microtubules, all these chromosomes arrange in the center. So here one chromosome, here below this another chromosome, right? So these are chromosomes and here centrosomes, then kinetochore microtubules. After stretching kinetochore microtubules, right? Then polar microtubules. So these are polar microtubules, right? Then after polar microtubules, what we should draw? Anchoring microtubules, right? So here, because of contraction of microtubules, these chromosomes arrange in the middle of the cell or equator plane of the cell or metaphase plate of the cell. This is center part, what we call metaphase plate. So all these chromosomes arrange in the middle of the cell. So how will you remember this? Metaphase. This is metaphase. Here, what is mnemonic? Meet in the middle. Who is meeting here? All chromosomes are meeting in the middle of the cell. So that's it about metaphase. Then anaphase. In anaphase, these microtubules start contracting. Because of contraction of these microtubules from this side, contraction of these microtubules. Which microtubules? Especially kinetochore microtubules. These kinetochore microtubules start contracting. When they start contracting, here centromere splits longitudinally. When they splits longitudinally, here this is sister chromatids. These sister chromatids becomes daughter chromosomes. So they splits and forms two daughter chromosomes, right? Let me draw that structure here. Cell, this is cell, right? Now this is one chromosome and this is kinetochore or what is it? Centromere. Then here another chromosome with centromere in the center. This is centromere, right? Now what is this? Centrosomes, clear? And here kinetochore microtubules, polar microtubules, anchoring microtubules. Here anchoring microtubules, right? See, because of contraction of these microtubules, what are these microtubules? Kinetochore microtubules. This centromere splits longitudinally. If you want to tell very accurately, here two sister chromatids held together by one protein called cohesin. What is it? Cohesin, right? So this cohesin splits longitudinally. Because of splitting of cohesin, these two chromatids separated. That means splitting of, if you feel it's difficult, just remember centromere, splitting of centromere longitudinally. Longitudinally means longitudinal to the chromatids. So here splitting and here splitting, right? So they have split and after splitting still there will be contraction, because of the contraction of these kinetochore microtubules. This half of the daughter chromosomes now, before sister chromatids, now daughter chromosome. These daughter chromosomes comes to this pole, this half of the daughter chromosomes will go to that pole, right? Once they reaches to their poles, then telophase will start. Up to this is anaphase. How will you remember anaphase? Like meet in the middle for metaphase. How will you remember? Here anaphase, apart. What is literal meaning of apart? Separate. So here separating. Who is separating? Sister chromatids are separating and becoming two daughter chromosomes. So apart. A for apart. Right? Then once they contract, these chromosomes comes this pole and these half of the chromosomes will go to this pole. Clear? Now, but I don't have much space, but still I will draw little smaller cell. I never mean that when keep on going down, the cell size will reduce. Of course, if you compare with mother cell, daughter cell size will be little less only, but not like this. Clear? Just imagine same size of the cell I have drawn here. Clear? Now, half of the chromosomes reached here, remaining half reached here with kinetochore microtubules I am drawing. This is kinetochore microtubule, this is kinetochore microtubule, and these are polar microtubules. Of course, centrosome, here centrosome, then anchor microtubules, anchoring microtubules. Clear? Now what will happen? I didn't draw remaining polar microtubules, so that's why I am drawing. Okay, so now what will happen? This is the last phase, telophase. So what is the process here and how will you remember? T for 2, T for 2. Here something is going to become 2. What is that? I will tell you after completion of explanation. This is reorganization phase. Whatever the processes which has occurred here, those processes will be reverse. This is deorganization and this is reorganization. So here aster formed. So here disappearance of aster. Disappearance of aster. So aster disappeared here. So after disappearance of aster, what will be there? Reappearance of nuclear envelope. So aster removed, then reappearance of nuclear envelope. Reappearance of nuclear envelope. How? Here I told you some membrane vesicle will be remained when nuclear envelope disappear. Those membrane vesicles united and forms nuclear envelope. So here nuclear envelope formed. How? By dephosphorylation of lamins. I told you here phosphorylation of lamins. Here dephosphorylation of lamins, which leads to reappearance of nuclear envelope or formation of nuclear envelope. Clear? Then reappearance of nucleolus. Reappearance of nucleolus. Right? Then here what has happened? Condensation of chromosomes, no? Here decondensation of chromosomes. Now we cannot see the structure like this, just we can see chromatin threads. Just chromatin threads. Right? So here chromatin threads. Clear? Now something became 2 here. What? 2 nuclei. 2 nuclei. So up to here, this phase, we have divided the nucleus, single nucleus into 2 nucleus, but not the cell. Cell still not separated. So till now we could separate nucleus only. So all these stages, what we call karyokinesis. What is this? All these 4 processes. All these 4 processes, what we call karyokinesis. Karyokinesis. Right? Clear? Only we could divide the nucleus up to here. Then what should we divide? Cytoplasm also we have to divide. That process what we call cytokinesis. Cytoplasm has to divide into 2. So actually there will be formation of one structure, one ring-like structure around the cell at the level of equator plane, otherwise at the level of metaphase plate. Right? So that ring, what we call contractile ring. What we call contractile ring. So that contractile ring will be formed from the anaphase itself. It will form from the anaphase itself. So there will be formation of one contractile ring like this. This what we call contractile ring. This contractile ring keep on contracting. Of course, this contractile ring is formed by actin filaments. So these actin filaments contract, so that just imagine one balloon with air. If you are holding the center of the balloon and keep on pinching, so that 2 balloons will be formed. In the same manner, this contractile ring will be contracting. So finally, this it will be formed like this. So finally this furrow, first it will form because of the contraction, it will form furrow. This furrow will be united and pinched up. This furrow will be united and pinched up. Right? So when it pinched off, cytoplasm also divided. Right? So now from one mother cell, 2 daughter cells has been formed. Right? So this is about cell division. Right? I hope you understood. Clear? Then once this cell divided, these cells are identical to each other and identical to mother cell. Identical to each other and identical to mother cell. That's why this division what we call equational division. Equational division. Equational division will occur in the somatic cells. Right? And another cell division is there. What is that division? Reduction division. That will occur in the sex cells. That I will tell you in the next step, next class. Right? Mitosis is somatic cells, meiosis sex cells. Right? Clear? Now once this cell completes its cytokinesis, then this cell enters into G1 phase again. Just imagine this cell only, this one, just imagine this one only, otherwise I will remove to avoid confusion. Now this is the cell, right? This cell reached here and entering into G1 phase. Once it enters into G1 phase, the size of the cell increases, right, and remaining growth of the cell will happen. Once completion of cell growth, right, then it enters into S phase, then G2 phase, then M phase, again it will enter into G1 phase, right. So this cycle will be going on happening in some cells, not all cells, right. So what are those cells? Example, cells which are present in the skin, cells which are present in the lining epithelium of GIT, right. All these cells will be going on, undergoes mitotic division. That means they will be always present within the cell cycle, they will not come out. That means there will be regular turnover, there will be torn cells replaced by another cells. If that superficial cells torn, again replaced by another cells. That means this division will be going on happening continuously. So those type of cells, what we call labile cells, labile cells, labile cells, labile cells, right? Clear? Then another type of cells. Now once the cell enters into G1 phase, it will increases its size. Then once it increases its size, it comes out of the cell cycle and enters into G0 phase, This phase what we call quiescence or G0 phase or resting phase, right. In normal circumstances, this cell will not enter into cell cycle, right. But if it get commands from higher center, that time this cell enters into cell cycle and divides. Once that commands are over, again this cell enters into quiescence. Let me give you example. Take liver cells. Once liver reaches the normal size and shape, the cells which are present in the liver, that means hepatocytes, they undergo quiescence. That means they will be resting in the G0 phase. In normal circumstances, they will not enter into cell cycle, right. But if there is any damage or if there is removal of half of the liver or some part of the liver and donated to other person, at that time these cells, hepatocyte cells, enters into cell cycle, completes the cell division, and they will be undergo cell division for many repetitions and stops cell division when liver reaches its original size and shape. Again those cells enters into G0 phase and there will be resting. Those cells what we call stable cells, stable cells, right, stable cells. Clear? Then another type of cells, once they enter into G0 phase, after completion of cell division, all these things, once they enter into G0 phase, they will not at all enter into cell cycle. They will not enter into cell cycle. Those cells what we call permanent cells. Permanent cells. Examples, neurons which are present in the central nervous system, right. So those cells are permanent cells. Actually, in the postnatal life, there is no cell division of neurons, right? Why? If there is cell division in the central nervous system, what will happen? Just imagine, today I came to your class, I took lecture about mitosis and I went to my home, and tomorrow if I come, if your neurons are replaced or undergone cell division, at the time totally you will forget and you will ask your friend that who is he, forget about my lecture, and you will not come to your college itself, you will go to some other college. Why it is happening like that? DNA replication in the neurons, it tends to decrease the storage of memory. It tends to decrease the storage of memory. Actually, if cell division is there, there should be DNA replication, is it or not? So, when DNA is replicated in the neurons, which tends to decrease the memory reserve, decreases the memory reserve. Clear? So, that is about different types of cells: labile cells, stabile cells, permanent cells. Labile cells going on replicating. Stabile cells, they will not undergo cell division in normal circumstances, but they will undergo in abnormal circumstances or in emergency circumstances, right? Then permanent cells, they will not at all enter into cell cycle. Clear? After completion of this, now we will see the checkpoints. Checkpoints means what? You must be aware of checkposts. When you are going to one place to another place, there will be checkpost. They will check your vehicle and they will check your license, RC, right? All the documents they will check, and they will check you also. If there is any gun, something if you have, they will check that also, right? For what? To prevent any damage, is it to the society or to any people? Of course, you must be aware of it. When you are going from one country to another country, there will be immigration checkpoints. There also they will check like this only. They will check your passport and they will check your luggage and they will check whether you are perfectly alright or not. If you are bleeding, they will not allow to enter. Clear? So, like that only, here some checkpoint is there. What is the checkpoint called? This is the checkpoint. This checkpoint what we call G1 and S checkpoint. G1 and S checkpoint. This checkpoint will checks and ensures there is no any damage in the DNA. If there is any damage in the DNA, it will not allow to enter into synthesis phase. It will send back again for repair. If there is repair, okay, again it will come and reaches to the G1 and S checkpoint. If that repair is proper, it will allow to enter into S phase. If not, again it will send. If the cell is going on coming to the checkpoint and going back, at the time this checkpoint will report to one gene called P53. That P53 gene will give some commands to that cell, either senescence or apoptosis. Senescence means old. That means that cell will not enter into cell cycle. That means no eligibility to enter into cell cycle. Right? It will remain in the body as a old cell or senescent cell. Right? Another one, if there is very severe damage, at that time P53 will send orders to kill. That means apoptosis. Right? So, cell it will die. Apoptosis. Clear? Right? Then, now after G1 and S checkpoint, after G1 and S checkpoint, the cell will go like this and completes its DNA replication, that means synthesis. Then here one more checkpoint it has to cross. This checkpoint what we call S and G2 checkpoint. This checkpoint will checks and ensures that there is no any damage of DNA during replication. During synthesis or during replication, no damage. Then only it will allow to enter into G2 phase. If there is damage, again send back. Clear? Another important one is here. Metaphase checkpoint. Metaphase checkpoint. This checkpoint will checks and ensures that all the kinetochore microtubules are attached to the kinetochore or centromere. Right? If one of the kinetochore microtubule not attached, it will not allow to enter into next phase. Why? What will happen if there is no attachment? Here unequal separation, unequal segregation. See here, if it is not attached, this chromatid will not go that side. It will come this side only. So that in this cell, how many chromosomes? 47. In this cell, one chromosome less. That means 45 chromosomes. Is it? So, to prevent that, this checkpoint checks and ensures all chromosomes are attached by kinetochore microtubules. So, these are the checkpoints. Clear? I hope we completed total mitosis. Clear? Next session we will discuss about meiosis. Right? Thank you.